Hi, this is Yogita. As a part of my assignment today, I'm going to explain about restriction enzymes. Restriction enzymes are also known as molecular scissors and molecular scalpel. Restriction enzymes falls into the subclass of nucleases that cut the DNA or the phosphodiester bond at specific sites, and they are naturally found in bacteria as a defense mechanism. For example, if we take the bacteriophages, which are the viruses that infect the bacteria, so the bacterial genome and the bacterial cells will be producing the restriction enzymes which cut the genome of the viruses so that the viruses will be killed, and it will be a major defense mechanism against the foreign DNA. Every restriction enzyme has their own recognition sequence and target site. For example, the restriction enzyme EcoR1 has the recognition sequence as GAATTC, and it cuts the phosphodiester bond between guanine and adenine. Based on the cuts made by the restriction enzyme, there will be two types of fragments which will be forming. That are blend fragments and cohesive fragments. And there are three types of restriction enzymes which are available. Type 1 restriction enzyme, they'll be cutting the DNA at 1000 base pairs away from the recognition site. And type 2 restriction enzymes will be cutting specifically at the recognition site. And type 3 restriction enzymes will be cutting 25 base pairs away from the recognition. In 1972, Smith and Nathan have discovered an restriction enzyme called as HIND3, and from there the nomenclature of the restriction enzymes have started. Usually, the first letter in the restriction enzyme will indicate the genus of the organism, and the second two letters will indicate the species of the organism, and the third letter will indicate the strain of the organism, and the Roman letter will indicate the order of discovery of the organism. For example, if we take a restriction enzyme EcoR1, Escherichia is the genus of the organism, coli is the species of the organism, and R is the strain of the organism, R13. That is, R13 is the strain of the organism, and 1 is the order of discovery of the organism. So, as we have already discussed, the restriction enzyme will be recognizing the DNA at a particular site, which is called as recognition site. At that site, the restriction enzyme will be cutting the DNA into fragments. Restriction enzymes cut DNA by breaking the phosphorester bonds in the sugar phosphate backbone. They leave sticky or blunt ends depending on the type of enzyme and the location of the recognition, and the resulting fragments can be separated by agarose shell electrophoresis. Restriction enzymes are used in gene cloning to form recombinants in which the plasmids are cut and the foreign DNA is inserted into the plasmid. They are used in DNA fingerprinting to identify individuals based on their unique restriction fragment pattern. They are used in genetic engineering to create transgenic plants or animals. For example, if we take Bt cotton, in which the gene from the Bacillus thuringiensis bacteria is inserted. So, because of the gene, the plant have become insecticide and pesticide resistant. By changing the amino acid sequence of the restriction enzyme, we can modify them so they can recognize the new sequences. Restriction enzymes are commercially available in the market. The price and quality of the enzymes vary depending on the suppliers and the type of enzyme. And there are various limitations of restriction enzymes. Some DNA sequences are not even cut by any known restriction enzymes, and some restriction enzymes have star activity, meaning that they cannot cut at the specific sites. They cut at the non-specific sites. Some restriction enzymes require special reaction conditions or cofactors. New restriction enzymes are being discovered and characterized at a rapid pace. The use of next generation sequencing and bioinformatics is facilitating the discovery of new restriction enzymes. Restriction enzymes will continue to be an valuable tool in molecular biology and genetic engineering. Thank you.